And but let's go ahead and talk about them here real quick. And what I'm going to do is I'm going to go ahead and divide up this lecture into two. This first one will deal with the structures, or with the organelles essentially. Right? So I'm going to go ahead and describe the organelles and the structure of them. The second part is going to deal with three cellular systems: right? The nuclear transport, the endomembrane system, and the cytoskeleton. Those three. So, you know, we'll go over the lecture here on these parts, and then these is going to be located in another, found in another lecture, a shorter lecture, maybe 20 minutes. All right, so again, okay, so now we should know by now, right? The two major kinds of cells that exist here on Earth. We have eukaryotic cells, right? And then we have prokaryotic cells. So these two domains of life, you know, deal with prokaryotes, so bacteria and archaea. Bacteria, you know what bacteria are. These are microscopic single-celled organisms that live in you, on you, and around you. Right? Archaea are also bacteria, but they're extreme bacteria. Okay, so these don't play around. Right? Okay, so now eukarya, right, is the other domain, and these right here are composed of, you know, first of all, they have organelles, right? So these have parts that are surrounded by membranes that are not found in prokaryotes. Okay, so again, the basic difference between the two is the fact that eukaryotes have a membrane-bound nucleus, and prokaryotes don't. Okay, so eukaryotes have membrane-bound organelles, and prokaryotes don't. So that's the basic difference between the two. Ready? So let's go ahead and talk about bacteria first. So this is a picture, well, of two pictures of bacterial cells. This one on the bottom is an actual bacterial cell looked at under an electron microscope. Okay, so this is an electron microscope image of a bacterial cell. Right, so in this one right here was recreated using computer models, evidently. So notice the few parts that bacterial cells have, right? They're rather simple. They have one plasma membrane, just one plasma membrane, which surrounds the whole cell. They have a nucleoid region, which is where their DNA is located in. And they have a plasmid, which is another site for DNA. This is another way for bacteria to store DNA. They're called plasmids. They have ribosomes. Ribosomes are made of RNA. They're not organelles. They're just parts made of RNA, and more specifically called rRNA, ribosomal RNA. They also have a cell wall that surrounds the cell itself. They have a capsule that surrounds the cell wall, and then around them they have other structures like fimbriae and flagella. Let me see if I have a picture. Fimbriae, these are hair-like structures right here. They have flagella that allow them to move, essentially. And flagella are most specifically for movement. Fimbriae are more specifically for attachment, right? So they can attach to other bacteria and also to surfaces. There's one more structure that's called a pilus, P-I-L-U-S, a pilus. right? That pillis allows for, it's called bacterial conjugation, right? Which is a form of, a form of exchange of genes between different bacteria. Okay, so those are, mostly the parts of a bacterial cell. Right, let me show you a couple of animations here so that you can see them in action, essentially.
The circular chromosomal DNA of bacteria must be compacted about 1,000-fold to fit within a bacterial cell. Part of this compaction comes the formation of new domains, where segments of DNA are folded into loop-like structures that are held in place by DNA-binding proteins. The formation of new domains compacts the DNA tenfold. Further compaction is a process known as supercoiling. This start introduced in the DNA molecule that causes it to compact to allow to itself. Coiling is observed by clustering of and looping and supercoiling make the bacterial chromosome complex within the bacterial cell.
All right, so that was an animation of bacterial DNA. Right? We talked about the tertiary DNA, the tertiary structure of DNA a few weeks ago. That also applies to bacterial DNA, but it's not called tertiary structure. That's basically called a chromosome compaction. Bacteria have one chromosome. They only have one chromosome that has genes in it. And in order to fit inside of the cell, they must compact it into a nucleoid region. Okay, so that's important right there. Plasmids are another DNA storage site for bacteria.
Bacteria have been helpful to genetic engineers, not only in providing them with restriction enzymes, but also with what are called plasmids. In addition to their single chromosome, many bacteria also contain tiny rings of DNA called plasmids. Plasmids are usually about 1,000 to 100,000 times long, and as independent, self-replicating molecular operators within bacteria. Plasmids are one of the ways that DNA combination goes on within nature. Bacteria's chromosome contains all the necessary genes for bacteria's existence. Plasmids provide to transfer plasmids contain genes that code for enzymes that digest certain antibiotics, such as penicillin or penicillin.
Okay, so here's what that means, right? Bacteria have another DNA storage site called plasmids. Right, so what we do nowadays with plasmids is we use them to engineer other molecules, right? So we use bacteria, for example, to make insulin for us. Insulin used to be made or used to be extracted from pigs, from animals before, but it turned out that insulin in people with diabetes, it caused a lot of problems. Therefore, nowadays, right, we're using bacterial plasmids to be able to, genetically engineer essentially the production of insulin. So in this manner, we use human insulin, and human insulin obviously will not cause the same problems that animal insulin causes, right? So what researchers do is they extract these plasmids from bacteria, right? They go and cut the plasmid at a certain point using what's known as restriction enzymes and CRISPR technology, right? So CRISPR technology was discovered by a scientist known as Jennifer Doudna, and she just earned a Nobel Prize, right? Nobel Prize for discovering these CRISPR technologies and using them in bacteria. Righty, so what you can do is you can get a gene from a human being, for example, the insulin gene, right? Inserted using CRISPR technology into these plasmids, and then insert the plasmids back into the bacteria where you got it from, and voila, you have your bacteria making insulin for you. All you got to do is collect the insulin, find a way to collect the insulin, make it into some sort of drug that you can sell to people, and sell it and become a millionaire, essentially, right? So there it is. And not only that, obviously, but you're contributing a lot to society. You're creating a relatively cheap drug that people can benefit from. So you're saving lives, aside from making a lot of money, right? So there it is. That's why plasmids are important. They're DNA storage sites. Ribosomes are another site within the bacterial chromosome, okay, which is also found in eukaryotes. Ribosomes build proteins. Okay, so they grab the mRNA that came from the nucleus, and they read that mRNA in three-letter codes called codons. So those three-letter codes, they represent an amino acid. So the ribosome assembles amino acids together using peptide bonds, right, to go ahead and create proteins. Right. So something else that bacteria have is they have a cytoskeleton. It's not as extensive as our cytoskeleton, but they do have a semblance of a cytoskeleton in them. Right. So what does the cytoskeleton do? Well, it maintains the cell shape. It maintains the shape of the bacterial cell. And it also plays a role in the moving of molecules from one place to another place inside of the bacterial cell. Okay, so some other bacteria, they can photosynthesize. Okay, therefore they're going to have some photosynthetic membranes that allow them to carry on with photosynthesis. We know what photosynthesis is. The way in which we define photosynthesis is the transformation of UV radiation into ATP and energy, right? Sugars for that matter. For not only plant cells, but some bacterial cells can also do that. So you have, some bacteria have photosynthetic membranes that can allow them to do that. Similar to chloroplasts, essentially, in plants. But plants are mostly exclusive for having chloroplasts. Right, so now let's talk about eukaryotic organelles now, right? Eukaryotic organelles have membrane-bound compartments. They're called organelles, membrane-bound compartments. What does this mean? Well, this means that every single part of the eukaryotic cell is, one, it's going to be surrounded by a membrane, and two, it's going to have a specific job. It's going to have a specific role within the cell, right? So let's go ahead and start with, or let me finish off rather, I'm sorry, with a bacterial cell wall before I move on to the eukaryotic organelles here. So here's the bacterial cell wall. It's right here in the middle, right? Bacteria have a membrane, which is found here, and it's also found here underneath the peptidoglycan layer. Right? When we talked about carbohydrates, one of those polysaccharides that I described was peptidoglycan. Lecture-Cellular is basically a layer of NAM and NAG cross links. Right? So those are carbohydrates. So some bacteria have a bacterial cell wall that is sandwiched in between two membranes. Right? And this is the plasma membrane right here, with the cell wall sandwiched in between. Other bacteria have the cell wall found over here on the top. So this one's called a gram-negative bacteria, and this one's called a gram-positive bacteria if they have a cell wall on the outside. And I think I already described this, the bacteria flagella, which allows for movement. But let me show more or less what that looks like.
The long whip-like portion of the flagellum is called the filament. Filaments are composed of many identical protein molecules called flagellin. Molecules of flagellin form identical chains around a certain pathogenic bacteria identified and classified by differences in their flagellar proteins. In contrast, the organic flagellum covers the filament of a prokaryotic flagellum. The whip-like filament inserts into a group structure called a hook. The hook is composed of a different protein and moves within the basal body. The basal body is composed of different proteins than the filament and hook. The basal body is composed of a rod and a series of other chloroform rings, which anchor the flagellum to the cellular wall and within its basal body.
All right, so it's a very complex structure right here. There's no captions here, so let me just kind of describe it in case you can't hear on your side. Here's the flagella, right? Let me use another. So this is the flagellum from a bacterial cell or a eukaryotic cell. Right? Most bacterial cells have a flagella because they can move. Not all of them, but most do. Notice how the flagella is anchored to the membrane, right? To the membrane structure of the cell. It's anchored through many different types of proteins and mechanisms that keep the flagellum in place. So what is the flagellum for? Well, the flagella is going to be moved in order to act as a propeller, essentially. So the flagella moves the cell. Some eukaryotes also have a flagella, like some fungi, for example, have a flagella. And then human cells, like a sperm cell, has a flagellum as well in order to be able to move. I can't think of another human cell that contains a flagellum other than a sperm cell. Other cells have a cilia, but cilia are not the same thing as flagella. They're similar, but again, cilia does not allow for the movement of cells. That's for something else. Okay, so now let's get into eukaryotes now. Again, eukaryotes have membrane-bound compartments, right? Otherwise known as organelles. Okay, so organelles are compartments essentially. Okay, so you've seen a generalized animal cell with all of its organelles, right? So there's the nucleus right there in the middle, the nucleolus inside the nucleus, right? The nucleus has DNA in the form of chromosomes. The nucleolus makes RNA. The nuclear membrane has pores, right, through which mRNA leaves. And the function of the nucleus is to store DNA, to go ahead and transcribe DNA into RNA to start the production of proteins. The function of the nucleolus is to create ribosomal RNA. Now, the animal cell also has this endomembrane system, which is made up of the smooth ER and the rough ER. The ER is the endoplasmic reticulum, right? So which we're looking at here, a set of membranes. The smooth ER creates some beneficial lipids for the cell. The rough ER contains ribosomes that are in charge of making proteins. So they make proteins for the cell. And then once proteins are made, they go ahead and travel through the extensive membranes of the ER. And proteins get folded, right? So we talked about folding of proteins. We have primary structure, secondary, tertiary, quaternary. All of them achieved right here inside of the endomembrane system. Okay, so next is a Golgi apparatus, right? Golgi apparatus right there, which has three functions. It receives proteins from the ER, sorts them out, and ships them out of the cell to be exported out of the cell. So that's the Golgi apparatus right there. There's peroxisomes, there's lysosomes as well, which carry on with recycling, right? So getting bricks that don't work anymore, killing bacteria, degrading lipids that can be harmful for the cell. There's mitochondria, right, as well, which produces ATP. Centrioles, part of the cytoskeleton, and the cytoskeletal system right there is all of these fibers, right? So those are part of the cytoskeleton. They're called microtubules, which move molecules inside of cells. Right? So again, these are things you've already seen in the past, right? So here's a plant cell, very similar to an animal cell, with the exception of the cell wall, right? So plant cells have a cell wall, which we're looking at here on the outside in yellow, right? Plant cells have a chloroplast, which carries on with photosynthesis. They have a central vacuole that stores nutrients in water, mostly, right? And these are things that differentiate the plant cell from the animal cell. Everything else is the same. Everything else is the same. A nucleus, nucleolus, ER, rough ER, smooth ER, mitochondria, peroxisomes, right? Golgi apparatus. Everything else is, in essence, the same. Evidently, there's a few differences, right, between the an animal cell. Now let's get into each one in particular, right? So starting with the nucleus, recall the nucleus stores DNA, and it also transcribes DNA into RNA. Right? So it stores DNA and it transcribes DNA into RNA. The nucleus is surrounded by an envelope, which is a membrane that has pores. And so it has pores. Once that RNA is made, RNA needs to exit the nucleus through the pores. And this is more or less what it looks like. Here's an electron microscope image of the nucleus. Here's an animation, right, with all of its pores right there. So we can see some pores there, right there, here. Right? Again, that allows for RNA to leave the nucleus once it's made. Ribosomes are next. Like bacteria, right? Eukaryotic ribosomes do the same thing. They regardless of the type of cell, it can be a bacterial cell, animal cell, plant cell. Ribosomes are always going to go ahead and assemble amino acids into proteins. That's their job, right? And then they go ahead and do so by creating peptide bonds. peptide bonds in between the amino acids, and that builds, the proteins essentially. Okay, so here we're looking at ribosomes, actual real ribosomes under electron microscope, and these are animations of ribosomes based on extensive research. Scientists nowadays are able to come up with three-dimensional views of ribosomes, given these two-dimensional images extracted from electron microscopes. Okay, so in electron microscope, in case you don't know what they are, those are huge, microscopes that can magnify thousands of times, essentially, thousands of times. Whereas the regular microscope that you've probably used in the past can magnify up to 100 times, right? So, it's obviously a very different, revolutionary type of microscope. We don't have one, right? Only research universities have these electron microscopes. UTEP has one, I believe, and one or two. And if you take a higher level biology class at UTEP or another university, you can take a class to learn how to use the electron microscope. Very complex stuff. Endoplasmic reticulum is another structure. It's divided up into two, rough ER and smooth ER. The rough ER has ribosomes, which we know what they do, right? We know their functions already. The smooth ER does not have ribosomes, and the smooth ER is going to go ahead and create lipids for the cell. It's going to go ahead and create lipids like cholesterol that are going to go ahead and become part of the membrane, essentially. So again, we've already talked about rough ER. Rough ER having ribosomes that create proteins. Smooth ER synthesize lipids. To synthesize means to produce, right? And these are lipids that are needed by the cell, like cholesterol, for example, but also by other parts of the organism. Alrighty, so under electron microscope image, this is what an actual ER looks like with ribosomes. This is a obviously computer-generated model, three-dimensional model of the actual rough ER. This is a smooth ER right here, which is basically another section of the same thing. So we're looking at the entire endomembrane system. One aspect of it is the rough ER. Another section of it is the smooth ER. So electron microscope and actual image and computer image right here. Okay, so now the Golgi apparatus. So three things that the Golgi does. One, receiving proteins from the endoplasmic reticulum, from the rough ER. Two, sorting out those proteins into different categories. And three is exporting those proteins to the outside of the cell, to places where they might be needed, right? So if you get an injury and you damage part of your tissues that need proteins, then you're going to go ahead and increase the rate of Golgi function so that the Golgi can export whatever proteins that you need in the injury site, right? And that way you can have those available to be able to repair your tissue, right? So that's just one extreme example, but the Golgi apparatus, it works all the time essentially, right? In creating and exporting proteins. The Golgi has two faces. It's got a cis face and a trans face. The cis face is oriented towards the ER. So this is the cis phase. It orients itself towards the nucleus and the ER. The trans phase is the opposite phase of the Golgi apparatus. Right? So the trans phase takes care of the export of these proteins, right? Now, here's more or less how that works. Once proteins are made inside the rough ER, they travel to the Golgi, right? The Golgi makes vesicles from itself, right? So these are little vesicles that are budding off of the Golgi apparatus, right? Hopefully you can visualize how that takes place. And these vesicles came from the Golgi by budding off of the Golgi membranes, right? So the reason why they do that is because they carry proteins inside of them, right, which diffused from the Golgi into the vesicle, and those are going to be for export. So those are the little sac-like structures that are going to export proteins. So actual electron microscope image and three-dimensional computer generated image right there. Let me see if I can show the animation of the Golgi.
It is important that translated proteins are delivered to their specific cellular location. To accomplish this, the protein is transferred through a series of membrane structures. The principal membrane component is the Golgi apparatus. The Golgi apparatus is the sorting organelle of the cell. Proteins from the rough endoplasmic reticulum are sent to the Golgi. As the proteins move through the Golgi apparatus, they are modified and packaged into vesicles. Because the Golgi apparatus receives proteins from one location and targets them for delivery to a second location, it is sometimes considered the post office of the cell. The Golgi apparatus consists of general components. The cisterna nearest the endoplasmic reticulum, or ER, medial and trans cisternae, the trans-Golgi network. Other key players in this process are the proteins being transported and the enzymes that modify them. Translated proteins are encapsulated in vesicles in the ER. A group of these vesicles fuse, and these fused vesicles form the cis cisterna. As the protein moves through the stack, it is modified by resident Golgi enzymes at specific locations in the apparatus. These modifications are important because they provide the signal that determines the final destination of the protein. So how does the protein move through the Golgi? Movement occurs in waves. First, the cisterna becomes part of the medial Golgi cisternae. Behind it, a new cis cisterna is formed by the fusion of vesicles from the ER. Meanwhile, one of the medial cisternae migrates and becomes the new trans cisterna. Collectively, this process is known as the cis maturation model. Proteins are sorted within the trans-Golgi network. Proteins with the same target sequence are destined for delivery to the same location. The trans-Golgi network then buds off into vesicles. These vesicles then migrate to their target location. These locations include internal organelles, such as the lysosome, the digestive organelle of the cell.
Right, so there it is. That's what I meant, by budding off of the Golgi. Right, so once they bud off of the Golgi, they're carrying proteins right there that are meant for export. Some of those proteins stay inside of the cell. Most of them are going to be exported out. All right, so lysosomes are structures that take care of debris that doesn't work anymore. Lysosomes are part of the recycling center, if you will, of animal cells, right? So if the cell has proteins that don't work anymore, then the lysosome takes care of them by dissolving them in acid. All right, so that lysosome has acid inside that dissolves those misfolded proteins that don't work anymore, as well as bacteria that might have invaded the cell. Okay, so here's an actual lysosome. Here, electron microscope, and here's a computer generated. And what do? Well, they take care of things that don't work anymore, and they also kill bacteria. All right, so all of these structures that I just mentioned, all of them are collectively known as the endomembrane system. Okay, so all of them are collectively known as the endomembrane system. Here's another structure called a vacuole. Vacuoles have storing roles. Right? So vacuoles are going to go ahead and store materials within them. I know I have a lot of stuff right here, but all you need to write about vacuoles is that they store materials. Vacuoles in plants are larger than vacuoles in animal cells. Plants have a central vacuole, right, that stores water mostly and some nutrients. Right, so mostly water and some nutrients. This is especially important in desert plants, right? Desert plants that need to store a lot of water, they must have a rather large central vacuole to be able to store all of that water and nutrients. Peroxisomes are a similar structure to lysosomes, but peroxisomes are more specific for lipid degradation. Alrighty, so peroxisomes are going to go ahead and detoxify cells by getting rid of lipids that are not needed by the cell. Right, so aside from other functions, peroxisomes are going to mostly detoxify the cell. Okay, so again, very similar to lysosomes, right? Here's one under electron, actually three, 1, 2, 3, under electron microscope, and that would be the computer, model of a peroxisome. So these and lysosomes are very similar, right? They help clean out the cell essentially. Okay, so next is the mitochondria, right? The mitochondria supplies ATP to the cell, as you probably already know. Okay, so how does it do it? Well, it does so through a process called cellular respiration. Cellular respiration, right? So cell respiration is, oh my goodness, again, it's divided up into three, sorry about that, into three main processes. Okay, here it is. So the mitochondria carries on cell respiration, right? Which is broken down into glycolysis, cycle, and electron transport chain. Those are three main processes. So as you consume sugars, as you consume carbohydrates, those carbohydrates travel to the cell after absorption, and they go ahead and make it into the mitochondria. So they're inserted into the mitochondria, right? Now, before they're inserted, they're broken down by glycolysis. So there's enzymes in the cytoplasm that break down carbohydrates, especially glucose. And that right there allows for the second process to take place, because these products are going to go ahead and travel into the mitochondria to undergo Krebs cycle and electron transport chain in the process of making ATP. So this is the most important reason why we have mitochondria. Right? It's to make ATP, which is energy, as we know. Right? So it turns out that mitochondria have their own DNA. Right? So after extensive research, it's been discovered that mitochondria have their own DNA. Right? So what does that mean? Well, that means, and they can also make their own proteins. So they have ribosomes. What that means is that was indicative of the fact that mitochondria used to be an independent bacterial cell, right, billions of years ago, about 3 billion years ago. There was this event called endosymbiosis, right? Endosymbiosis that allowed for mitochondria to become part of prokaryotic cells. Right? So endosymbiosis allowed for mitochondria to become part of other prokaryotic cells, ultimately interacting with them positively to make the eukaryotic cell as we know it nowadays. Right? I hope that makes sense. Now we'll cover that later on in more detail. But again, endosymbiosis 2 to 3 billion years ago allowed for the modern eukaryotic cell to be generated, essentially. Right? So there it is. Now the chloroplast, right, is the equivalent to the mitochondria bit in plant cells. Okay, so now plants don't eat, right, like we eat carbohydrates. Plants make their own carbohydrates. But in order to do that, they have to absorb sunlight, right, first. So that's what the chloroplast is for. The chloroplast, excuse me, the chloroplast is going to be divided up into two parts. One is called the thylakoid, and the other one is called the stroma. The thylakoid and the stroma. The thylakoid is responsible for the green pigmentation in plants. Ready? So what they're going to do is they're going to go ahead and use chlorophyll to absorb sunlight, right? To carry on with part of photosynthesis. It's called the light reactions. Okay, so the other part, the stroma, is going to carry on with the second part of photosynthesis, which is called the Calvin cycle. Okay, so the stroma is going to go ahead and make the sugars that the plant needs from photosynthesis. Right, so again, the chloroplast is also an endosymbiotic organelle. It was also a bacteria once, an independent bacteria once, 2 to 3 billion years ago. But its relationship with other simpler prokaryotic cells also led to the formation of these plant cells. Again, the theory is called endosymbiosis. So here's a look at a chloroplast in reality and in animation, right? So these are called thylakoids. These pancake-like structures, and they're thylakoids. And everything else is a fluid, essentially. It's called the stroma of the chloroplast. Right, so again, what does the chloroplast do? Well, it's going to go and absorb sunlight, be able to transform sunlight into usable energy for the cell, ATP, and sugars for that matter. Okay, so last but not least is the cytoskeleton. The cytoskeleton, right? Let's break down the word here. Cyto means cell. Skeleton refers to some sort of structure. Right? Like, for example, your skeleton is made up of bones. Your bones give your body structure. Right? So the cytoskeleton is essentially the network of proteins that give cells their structure. So there's three types of proteins. There's microtubules, intermediate filaments, and microfilaments. Those are three types of proteins that give cells their shape. Okay, so another function of the cytoskeleton is movement of molecules inside of the cell. Right, so transporting materials within the cell itself. Okay, so now these images are not electron microscope images. These are fluorescent microscope images. Okay, so fluorescent microscopes. So what we're looking at here is the cytoskeleton being stained. So they're stained, looked at by using fluorescence, right? So notice how extensive. This is one cell, by the way. This is one cell. There's another cell, another cell, another cell, and so on. So 1 cell, 2 cells, 3, 4, 5. Notice how extensive their cytoskeleton is, right? It's very big and it comprises most of the cell. So cytoskeletons, again, are made up of microtubules, which we're looking at here. So microtubules are made up of proteins called tubulin. So tubulin proteins make up, a helical microtubule. There's another protein. It's called actin. Right? So these are intermediate filaments that are also part of the cytoskeleton. Right? So these are called actin. As a matter of fact, your muscles are also made up of actin molecules, right? So these actin molecules are found all over your muscles. They allow for muscle contraction. Right? So last animation here, you guys, and we'll call it a lecture. It's not now.
If you take a look at most drawings of a cell, a major component is usually missing, the cytoskeleton. Welcome to Moo Math and Science, the cytoskeleton. The name implies it is the skeleton of the cell. Well, kind of. Welcome to Moo Math and Science, the cytoskeleton. As the name implies, it is the skeleton of the cell. Well, kind of. The cytoskeleton of a cell is a network of filaments running throughout the cell. The cytoskeleton has several different jobs. First, it helps provide support, much like your skeleton provides for use. It provides traction or height for the movements of vesicles and other objects in the cell. It helps pull the chromosome apart during mitosis. It even helps the cell. Some single-cell organisms have cilia made up of cytoskeletons. The cytoskeleton consists of three types of fibers: microfilaments, intermediate filaments, and microtubules. Microfilaments are fine thread-like proteins and fibers. There is a most predominantly a protein called actin. These filaments create surface alloys vesicles that in the cell, and may even hold organelles in place. Intermediate fibers are medium and linear. They help maintain the shape of the cell. For example, a red blood cell has a uniform shape. Cytoskeleton filaments help produce this unique shape. And finally, microtubules are the largest filaments and also create tracks for proteins. Think of microtubules as roots in the cell. They also help pull the chromosomes apart in mitosis. Microtubules also help with the movement of the entire cell. Like the flagella or cell that helps prevent certain diseases. If you'd like to learn more about cell.
All right.